So let's start understanding how we get rid of weeds, how we set the stage to grow the plant you want to grow. So when you take a sample of your soil to begin with and trying to determine whether that's what you want or not, what do you need to get back into that soil? Well, you need to know what plant you want to grow and what does that plant require. So back at Colorado State University, where I was doing my PhD, postdoc at the NREL with Dave Coleman, and we did an experiment where we looked at the biology in all these different systems. And so irrigated wheat, this is the least productive system. Because they're using herbicides, they aren't producing a lot of plant material. Yeah, they're growing a decent yield of wheat, but that's not the question we're asking here. So the least productive system, the least amount of plant production in that system. Completely, strictly bacterial dominated. Cannot find a fungal hyphae in the system. Not a lot of protozoa. Not a lot of good guy nematodes. A fair number of bad guy nematodes. Pretty good set of root-feeding nematodes in that system. So we don't have the nutrient cycling. They are dependent on the inorganic fertilizers. No question that the inorganic fertilizers will work, absolutely none, but it is not the wisest use of our resources to go that direction. So going to dry land wheat, where basically they're just going to put a furrow, drop the wheat seed in, and cross your fingers and hope that God sends some rain, that there'll be a good summer, and you'll be able to get your $80 an acre back. A lot more fungi in that system, a lot more production. We do have weeds and typically some other short, low-growing understory plant species starting to develop. So a little more fungi in there, a little more productivity in terms of total plant production. Moving on to the short grass prairie on the Front Range of the Rocky Mountains in Colorado. A lot more fungi in that system, a lot more productivity. Huge diversity of different kinds of grass species, different kinds of plants. Typically, pretty much all of that surface is covered with plant and protecting the soil beneath. Quite a bit more fungi in that system. Tall grass prairie. In the years that we were working here, the tall grass prairie was getting enough moisture that the grasses were about 12 feet high. And so you're out there working through a summer's day and, taking soil samples and, our ice chests and getting all ready to go home at the end of the day as the sun is sinking into the western horizon and the coyotes are starting to howl. And you turn around and you go, where's the car? All there is 12-foot tall grasses around you. There were a few moments when I thought the coyotes were going to win. Kind of scary. So we learned to leave bread trail crumbs. So tall grass prairie, a lot more productive, obviously. So ranking these things on productivity. And if you recognize this, we're also talking about succession. The earliest successional system, a little bit more further along. High mountain meadows up in Dryland Park, Fox Park in Wyoming, up in the Rocky Mountains, using how many pounds of weight gain per animal per day of the cattle and the sheep grazing this property, using that as an estimate of productivity, way more productive. Definitely on the fungal-dominated side. So the first time ever anybody publishes this kind of information are these papers that appeared in 1987, 1989, in that time period. Lodgepole pine forest, of course, very fungal. We have 1,000 times more fungi than bacteria in that soil. So productivity, successional gradient. So using this kind of information, can we suggest to you what the ratio of fungi to bacteria you might need for growing what kind of crop? Well, what stage of succession does your crop, where does it come from? What stage of succession do you find it in? So let's keep going a little bit, because this is the first time ever, when somebody wants to say, Elaine's never published any data about this, it's like, what, you can't read? Because it's there, troops. It drives me crazy when people say, Elaine's never published anything on the soil food web. Really? I have over 100 papers in the scientific literature. So when somebody says that to you, just inquire with them if they can read. So if I'm very bacterial, they're very fungal. The first time ever we demonstrated the most rapid rates of decomposition are in the winter under the snow. One of the things that occurs in the winter under the snow is if you have any decent fungi in your soil, you're going to have all of that plant debris, the litter, the residues that went down on the surface of your soil, they're going to decompose through that winter time period. If the snow melts in the springtime, and you still have a bunch of dead leaves, you still have a bunch of dead material, residue sitting on the soil surface, nature is trying to tell you you don't have any fungi. Perhaps it's time to get the fungi back into that soil, unless you really like growing weeds. So, starting to understand some of these things in the first year that Soil Food Web Incorporated started working, we worked in all of these different kinds of systems. All of these systems have the data about the food web published. It's just that it's not in a single publication. There'll be a publication about the different kinds of bacteria, and then another one about the disease-causing bacteria, and then another one about the pseudomonads, and another one about the bacillus species. And then there'll be a paper about several different types of the fungi in the system. And then there'll be another paper about the flagellates and amoeba, another one about the ciliates, another one. So if you really want to find the information on rhododendrons in North Carolina, you're going to have to go and collect about 150 papers from the scientific literature. And I'm perfectly happy if you do that, or maybe we could save you a little bit of time if you just come and talk to me. Because most of this data sits right there, because it makes a lot of sense. The overwhelming framework is very clear. So agricultural systems that we worked with in the first couple of years at Soil Food Web, we work with over 3 million acres of agriculture, landscape, gardens, so lots of data about lots of things. So to exemplify the kind of work we do, we're going to go into a watershed, because this is all on the same parent material. So we don't have to be fussing about, we're changing the sand, the silt, the clay, the base mineral material that is present in these different systems. So going to the old growth forest, we're looking at the micrograms of bacteria per gram of soil, so this is total. There we go, there it is. We're going to look at the micrograms of fungal biomass per gram, and this is total fungi. And so there you are. And so you can see that in that old growth forest that we have more than 2 times more fungi than bacteria. And that is always going to be that kind of ratio. 2 to 5 to 10 to 1,000, depending on how healthy your old growth forest actually is. So I take a look at this data, and I'm kind of worried about this old growth forest. It's really not very healthy. When we go to the most productive ecosystem on this planet, and we start looking at the fungal biomass in that ecosystem, one single individual fungus is 20 miles across, and it goes down into the soil at least 25 feet. In the springtime, when the snow melts, anything you pick up from the forest floor is all held together. You can't just easily take a handful out of the soil, because you're ripping and pulling, and you stand there with, it's kind of like wind chimes or something. All this soil is held together with that fungal hyphae. Nice. Well, sometimes we look at some fungi that are pink, some that is orange, white, black, brown, lots of different flavors. But this largest organism on the planet is 20 miles across and goes down at least 25 feet all the way through that part of the forest.
And where is that?
Where is that? Where is the most productive ecosystem on the planet? It's up in Washington State, in the Olympic Peninsula, in the cedar old-growth forest. And so Paul Stamets is the person who's been doing the work on this particular fungus, using DNA analysis to demonstrate that it is indeed the same individual all the way across. So come springtime, snow melts, every drop of nutrient being pulled inside that fungal hyphae, being retained, not being lost from the system. If it's a healthy forest, nothing but clean water is coming out the bottom of that soil. But then, of course, the predators wake up. So the flying squirrels wake up. And oh, do they start having a great time chowing down on that fungal hyphae. The microarthropods, the earthworms, the voles, the shrews, the mice, the fungal-feeding nematodes, and it's just this great big party in the below ground in these systems. Think about the nutrient cycling. As all that massive amount of fungus just gets chewed up, releasing all those nutrients, and the plants, of course, are saying, thank you very much. Perfect. Hey, guys, I'd like a little more cobalt, maybe a little more boron over here. And so the plants are getting precisely what they need. So come late July, early August, all you're finding is a little clump of that fungal tissue, maybe another little clump, and you go and people go out into the old-growth forest at that time and they go, no, we don't have 20 miles across fungal hyphae, because somebody's eaten them. But then fall comes along, all that litter, needle litter falls to the surface of the soil, all the understory plants, all that residue on the soil surface, snow falls, the fungi start to regrow and reestablish 20 miles across, 25 feet down. So think of the nutrient cycling. Why is that old-growth forest the most productive system on this planet? Look at the nutrient cycling. Yeah. So round and round and round it goes. And so productive because of the biology present in the system. So not a really healthy forest here. This should be way higher, but that's what was in this system, fairly human impacted. Pastures, just a little bit down from the crest where the old-growth forest was, 2 pounds of weight gain per animal per day. We're looking at still fungal dominated in that system. And yes, look at that, the bacteria are actually higher earlier in succession because we don't have the fungi competing with them. So the fungi help suppress those bacteria in that old-growth system. So when we're getting into that highly productive pasture, 2 pounds of weight gain per animal per day during a 90-day feeding study is pretty good rates of weight increase. We've worked with a lot of people in Texas where we get up to 5 pounds of weight gain per animal per day if we get the biology right. So get it right and you can get these higher weight gains. How can you be gaining more weight? Just because they're eating some grass? Well, think of the nutrients. The grasses that we're dealing with here were about 18% protein as compared to here where they're only 5% protein. Would you grow faster if you're a young growing calf or horse or cow or whatever? Would you grow faster if you had better food to feed on? Yeah. So what happens as we lose productivity within that system? Now do you know why? We're not getting the weight gains. The animals aren't getting the nutrition that they require because we've lost our fungi and we've lost a lot of the bacteria. How do you get this pasture back up to that place? You got to put compost in, compost teas, compost extract. You want to put the biology back into the system, especially that fungal component. And you might think about putting in some fungal foods just to, give the fungi a kick in the butt. Come on, guys, get growing. Do your job. Do that nutrient cycling so your plants, your grasses, your forbs, your herbs are going to have the nutrition in them so your animals can get that benefit. An agricultural field, 180 bushels of oats being produced versus 100 bushels of oats being produced. And again, equal biomass of fungi to bacteria. That's what we need for these highly productive row crops. So wheat, oats, barley, corn, soybean, equal biomass of fungi to bacteria pretty much. What caused the reduction in productivity, where we're only making 100 bushels per acre? Yeah, the pesticides they're using, the herbicides, are killing the biology in the soil.
What's the index?
So what is this? We have all of these courses online. So Life in the Soil is a 3-day course. It's about 48 lectures of 45 minutes each. Compost, so about 18 lectures, 45 minutes each. Compost tea, compost extract, and the microscope course. So those 5 courses are now being offered online. I'm amazed by people that come to a talk like this, and they'll say, Elaine, I watched that lecture 45 times. Whoa. Until he got it. Absolutely. So that's why I think the online course is really great.
Do you ever have anybody say that so much compost in the soil will kick up the potassium levels too high?
That's not compost. That's manure. Okay, so you have to compost manure properly. Any organic material needs to be properly composted. If you've gone anaerobic quite often, if you had high levels of potassium, phosphate, a lot of those different kinds of things, you're going to get a lot of soluble. Those things are going to be very soluble. If you go anaerobic, then you lose them all as gases. But those nutrient imbalances are because you have not properly composted. So why is it that we're getting the highest rates of decomposition right at the surface between that snow layer and the soil? Well, the snow is a blanket. It's a buffer. It's insulation. So it also means we're going to have free water right at that layer. You may well be completely frozen above. You may be completely frozen below. But at that interface, we have free water, and the bacteria and fungi are going gangbusters because the protozoa are asleep. The nematodes, microarthropods, the earthworms, there's nobody chowing down and keeping those bacteria and fungi controlled. And so they are just going through the whole system. And actually in the Olympic Peninsula, the soil does not freeze, even down to 4 or 5 feet. And so that fungus is going to completely regrow and the most rapid rates of decomposition.
There's a market gardener up in Quebec named John Martin Fortier, and he discovered by accident that if after he finished a crop, he laid silage tarp on it, it composted very nicely and the soil was nice and fluffy and everything. Is that silage tarp performing the same function as the snow?
It might be. So he's describing a silage tarp over the top, so your residues go down on the soil surface and you protect that perhaps. And if it's preventing ice crystal formation and you got free water.
Growing season, it's not in the winter.
Yep, so growing season, I'd really want to look at what's going on with that biology, understand that a little bit better. But a silage tarp is going to be preventing evaporation so that soil surface doesn't dry out. How do you prevent microorganisms from growing? The easiest, simple way to prevent microbial growth is to dry it out. Just like you and I. If we completely dry you out, are you going to be functioning? So they don't either. So prevent that from drying out, especially the soil surface.
He's also very insistent that it be UV protected. So I don't know whether that's just so that the tarp doesn't fall apart quickly, or whether the UV would have some effect on.
UV doesn't usually penetrate more than 1 millimeter. So UV would be negatively impacting the top 1 millimeter of your soil. So it's like, would it be enough in any situation to be a problem? Don't know. But so the plate index, go back to the question. After 20 minutes, the plate index here is where there were people that were using the growth of microorganisms on 5 different kinds of petri dishes. And I don't know if you've seen like a plate where we have a particular medium inside that little plastic dish. The medium in there is very limited. So, for example, with potato dextrose agar, where people have grown total fungi present in your soil for a very long time. How many food resources are there in a potato dextrose agar? Potato, which is starch. One kind of food. Dextrose, sugar. There's only two food resources in that we're going to grow all the fungi on the planet on starch and dextrose. Excuse me, most soil fungi do not use starch, nor do they use dextrose. So we're going to miss 99.999999999% of the fungi that are actually present in that soil. And then think about on that petri dish, we only have a very limited set of foods. So of course, we're going to miss most all the different kinds of fungi present in there. And then we're going to take, after we spread our sample out, we're going to put a lid on that dish. And we put that dish into an incubator. One temperature, one moisture, one humidity. How much are we reducing the number of species that can grow in those conditions? And then think about what happens on the plate as those microorganisms start growing in this atmosphere that's about this thick. In about an hour, what's going to happen to the oxygen in that atmosphere? Depleted. So what kind of fungi are we actually growing on that plate? The diseases, the pest fungi, because the beneficial fungi require aerobic conditions. The bad fungi, the diseases, the pests, are all things that require reduced oxygen. Now, there'll also perhaps be some good guy fungi in there, but we are really selecting for the diseases and the pests. So what's actually growing on those petri dishes? Yeah, so interpret this data properly. Old growth forest. Nothing grew on all 5 different kinds of media. Absolutely nothing grew on any of those plates. The laboratory that was doing these kind of plate indexes actually called the guys up. I didn't send the samples into that laboratory because I have a bit of an attitude about petri dishes. So they sent them in there, and the laboratory, this is in Tempe, Arizona. They called these folks up and said, so is this desert soil? and the laughter going on here is just about the same as the laughter these guys said, because they said, would you just go look at the soil in the bag? Good, rich, lots of humus, nice dark color to it. That's not desert soil. Well, why didn't any of these organisms that were present in that old growth forest, and wow, look at the number of organisms, why didn't they grow on any of the 5 different media? What temperature are we incubating at? 72 degrees. When's the last time that old growth forest soil was 72 degrees? About 800 years ago. So maybe you have to pay attention to the ecosystem in order to properly assess this. So, the highly productive pasture. 5 different species. One not as productive? Well, yeah, because we got a few more diseases in here. Take a look at that. Does that explain why this is not productive? Because look at the disease. Look at the problem organisms we've got in there. So petri dishes, learn to interpret that information correctly. It's not that petri dishes, it's not that that approach is bad. You have to interpret it properly. Interpret it as, look at the diseases we've got. Useful information in its own way. If you want to know how many E. coli are present in any material, you use a petri dish approach. Because we know the media, we know the conditions that E. coli grows in. We can resuscitate E. coli even if it's just that far away from death. We can bring it back. We know how to grow E. coli. So if you want that kind of information, petri dishes are the way to do it. But if you want to know all the bacteria, all of the diversity, you got to use this microscope method, the shadowing microscope method, because there's no other way to get everything and do it in a short period of time. So this is my slide just to prove to you that I have lots of data We'll measure the volume multiplied by the density of that material, so we're comparing biomass. And it's the same sort of thing where we're looking at that gradient. This is all in the same watershed, from total weed field up to old growth Douglas fir forest. And we can look at active and total bacteria. And notice that total bacteria increases to about midway through succession and then levels off. It does not keep increasing as we get into those late successional systems. Total fungi. Yeah, it's kind of bouncing around a little bit, but now look at it take over. As we're moving into those later successional systems, our fungi are just taking off. So if we're looking at that ratio of fungi to bacteria, we can get a really good idea where we are in succession. What are we setting the stage to grow? If we can then manage that ratio of fungi to bacteria, we should be able to instantly set the stage to grow whatever plant you want to grow. You don't want weeds? Get rid of them by moving you this direction. How are you going to get the fungi in there? Well, that's what your compost is for. Compost that we're going to use a lot of woody material in the compost pile, because wide seed-in ratio material is fungal food. What if you have no fungi in your compost? Well, then good luck. There will not be any decomposition of that woody material, and oh, you will have problems. If you have nothing to decompose that material, it's going to cause problem with your plants. So if you can't get those wood chips, if you can't get those leaves to decompose, nature is sending you a message. You don't have any fungi. Where are you going to find them? Go out into these old growth systems. Take a pinch, where you can see really nice fungal hyphae. Take a pinch from there, that goes into your compost pile.